video is going to be showcasing some modeling of DNA replication. So I'm hoping that by watching this video you'll be able to see this in more of a three-dimensional kind of way, better than just the labeling and the coloring or, and watching a video. I'm hoping that this might kind of clear up a few things. So what we have here is our molecule of DNA. It is double-stranded, and it's made up of 4 different nucleotides with 4 different bases. So the little letters on there, A, C, T, and G. A is, of course, adenine, T is thymine, C is cytosine, and G is guanine. And you'll notice they're different colors. And you'll notice the base pairing that's occurring. C and G are always together, which is blue and green, and then A and T are always paired up across from each other, and that's yellow and red. And so even though we don't see this spiraling, we can tell that it is 2 strands of DNA, and that those bases are pairing up, keeping that base pair rule in mind, A with T and C with G. So DNA replication occurs in the nucleus, and this is going to happen before mitosis happens. It happens during the S phase of interphase, and here we go. So the DNA first basically encounters an enzyme called helicase. And the helicase is the enzyme that unzips the DNA. So it is going to separate that DNA, and each strand on this picture is going to go through a different kind of path. But basically the job of helicase is to unzip the DNA, and that is the first step. Okay, we see our DNA has been unzipped by the helicase. And now we see the enzyme DNA polymerase has arrived. And the DNA polymerase is the enzyme that actually adds the DNA nucleotides to the original strand. So you notice there's 2 of them, and they are have arrows on them. They're showing that they're going to be moving in different directions. And this is because the DNA strands are anti-parallel. You can see those arrows on the DNA kind of pointing in opposite directions. And although this feels awkward, they're going to move along the strand in opposite directions. So the one that's on the top of your screen is going to be the leading strand, and that's because it's going to start at one end and do the whole section of DNA. And then the one on the bottom, you can see there's just going to be only a few nucleotides added before it kind of runs out of room. And then this DNA polymerase is going to have to kind of scooch off and then come back around and catch the next segment. So that bottom part is called the lagging strand, and those are going to be in short little sections. So it's a little less efficient than the top part, which is the leading strand. And so those nucleotides that are added, again, are added based on the base pair rule. So if I see a cytosine right here, I know what has to be added is that guanine. And when I see the adenine right here, I know what has to be added is the thymine. Okay, and that's going to happen simultaneously in both of those DNA polymerase kind of bubbles. Okay, so both of those are going to occur. I'm searching for what I want. There it is. And again, this is going to move along this direction, and this one's going to move along in the opposite direction. Okay, so as you can see, our DNA replication has gone a little bit further along, and so we can start to see that second strand be created on both of the original strands. You can see the top part of the screen is showing the leading strand, and that's kind of all in one piece, whereas the bottom one is going to be in short little sections, and again, that's called the lagging strand. So one more clip, and we will have our finished product as these DNA polymerases continue to add nucleotides until the end of their section of DNA. Okay, here's our finished product, and we have 2 new DNA molecules, and you can see one side of the DNA is that original colorful side that we had, and then the other side is the gray side, and those are the new strands, the new nucleotides that have been added. But if you look with me, you can see that they are identical. So see how on the first pair it's a G and a C, and on the left hand DNA and the right hand DNA are exactly the same. And if you look at that and follow that sequence, it is identical all the way down. Even though we see different colors here to show the old and the new strand, the sequence has been conserved, and that is the key to DNA replication, because the sequence of DNA, which we're going to see in our next set of modeling, is what makes the protein the kind of protein it is. So it's very important that when DNA is copied, that the sequence is conserved or kept, because otherwise we would be making all kinds of crazy proteins. So this process does need to be completely precise and needs to keep that sequence of DNA when it is completed. Now, this looks like we do the DNA from one end all the way to the other. And the truth is that DNA replication occurs at lots of different locations on the DNA all at once, so the process can be shorter. And that's DNA replication.